We can go ahead and get started this morning. It's my pleasure to introduce Dr. Bharat Thyagarajan. He is an associate professor here at the University of Minnesota in the Department of Lab Medicine and Pathology. Bharat did his medical training in India at Grant Medical College. He then decided to move to Minnesota to pursue graduate training. He received both his MPH and PhD in epidemiology from the School of Public Health at the University of Minnesota. He then continued his training here by doing both a residency and fellowship in the Department of Lab Medicine and Pathology, where he subsequently joined the faculty as an assistant professor in 2007. In 2012, Bharat became the medical director for the Molecular Diagnostics Lab and also the University of Minnesota Clinical Genomics Core, where he's done a lot of work developing genomic testing for clinical applications at the university, and that's what he'll be telling us about today. In addition to this clinical work, he also has an NIH-funded research lab where he focuses on discovering mitochondrial biomarkers for breast, colorectal, and hematologic malignancies. And in addition to that, his lab also provides support for a large number of multi-center epidemiologic studies. So we're thrilled to have him here today, and I will turn it over to Bharat.
Thank you, Jen, and thank you for inviting me to this talk and give me an opportunity to present my work. So today I'm going to be talking about the clinical applications of genomics in pediatrics. And I'm going to be focusing really on the work that we have done in the Molecular Diagnostics Lab with regard to next-generation sequencing and the application of large-scale sequencing in clinical diagnostics. So as an outline, I will describe the infrastructure that we have developed for clinical genomics here at the university. And then describe a large portion of my talk will revolve around the application of next-generation sequencing for inherited diseases, because that is the area where we receive a lot of clinical samples from the Department of Pediatrics. In addition, we have a robust genomics platform for diagnosis of oncological disorders. So I'll touch on that. And lastly, I'll finish up by potential for translational research opportunities that can be leveraged using the infrastructure we have developed really for clinical work. So as a brief background of what the molecular lab does, the molecular lab is situated in Mayo, and we process around 25,000 specimens annually. The major categories are listed below here. A big chunk of what we do, I would say 60–70% of our test volume is really driven by HPV or Pap screening and bone marrow engraftment analysis. Those are the tests that pay our bill. But I'm not going to talk about those things. I'm going to talk about the three categories down below, the heme and solid tumors, as well as the inherited disorders testing that we do. Because these are the areas that really benefit from us having next-generation sequencing technologies. So when we talk about the current paradigm of testing for any complex disorder, be it inherited or oncology related, the problems are similar in that increasingly genetic diagnoses is becoming more and more complex, in that there are several dozens to thousands of genes that are involved in getting to a proper diagnosis, a genetic diagnosis for any disorder. And in addition, you need to be able to detect not just single point substitutions, but a variety of different type of variants such as deletions, duplications, translocations, so on and so forth. And our current paradigm where, we think of a single test, a single mutation as a single test, has already changed. I used to be able to say that, we are in the process of changing, but at least in the last couple of years, I would say this paradigm has already changed. And increasingly, comprehensive diagnostics requires ability to detect different types of mutations, different types of genetic alterations, all from the same assay. So this is where we decided around probably now 6 or 7 years ago that we will make an investment in developing next-generation sequencing based diagnostics. The appeal of this technology was that, at least theoretically, it would be able to detect every conceivable type of genetic alteration that we could think of. And the tools that were available to us came in 2 different flavors. One is your industrial strength large-scale sequencing sequencers called the HiSeqs. And then you had your desktop sequencers. The difference between these two is really in throughput. Your desktop sequencers gave you roughly 10% of the data, of the sequence data you can get from the large-scale sequencers. The advantage is that the desktop sequencers gave you results in 24 hours. These instruments ran for a week to 10 days to give you a result. So they both had their advantages and disadvantages. So to utilize next-generation sequencing, we really started this work back in 2010. And we developed this clinical genomics center. It was a collaboration born out of necessity, but has existed till date, mainly because it's still the most cost-effective way to provide sequencing capability here at the university. So in 2010, what we did was, when we were thinking about how to develop this capability in next-generation sequencing, these instruments cost roughly $1 million in capital investment. And we, frankly, just did not have the money to do that. So I approached Kenny Beckman at the University of Minnesota Genomics Center and asked him if there was any way we could collaborate to do next-generation sequencing, but for clinical applications. And Kenny's Genomics Center was just getting started on next-generation sequencing for agricultural and plant veterinary stuff. And he said, oh, that sounds like a good idea. And he said, okay, we'll get CLIA certified so that we can do this for clinical diagnostics. And that's how this whole thing got started. And then we also approached the Minnesota Supercomputing Center to provide bioinformatics support for all the sequence data that we were going to generate. So this group of 3 units really have been operating since 2010, and these form the core of the infrastructure for all the work I'm going to describe today. The initial support for this clinical genomics center really came from the Institute of Translational Neuroscience. So Harry R. provided us some initial seed funding to develop our initial assays back in 2010. And subsequent work has been funded, especially in hiring bioinformaticians, upgrading HiSeqs, has been, and providing seed money to develop these assays further, been really was funded really by the Department of Lab Medicine and Pathology, Fairview, and the Bob Allison Ataxia Research Center. And more recently, as of January of this year, clinical genomics really has been identified as a strategic growth priority within the Fairview system. And so Fairview, through Fairview Lab Administration, has provided around $7 million over the next 5 years for us to develop at least a regional reference lab capability in clinical genomics. And I'll talk about what we intend to do with this investment that Fairview is going to make towards the end of my talk. But that's the evolution of how the infrastructure has developed here at the university. It's been close to 7 or 8 years of work to get to where we are at this point. So I'm now moving to the next part of my talk, which is really to talk about what we do for sequencing for inherited diseases. For inherited disease sequencing, we use the large-scale industrial strength sequencers that are located at the University of Minnesota Genomics Center. So the workflow is the Clinical Molecular Diagnostics Lab gets the patient sample, we extract DNA, we do all the library preparation, the pre-sequencing work, and then the sample is sent over to UMGC for sequencing. The raw sequence data is then sent over to the Minnesota Supercomputing Institute, where they spin off the data into an Amazon cloud. We have a pipeline that is set up for analysis of data. And then the Clinical Molecular Diagnostics Lab pulls the process data from the cloud, analyzes it, interprets the variants, fills in any gaps with Sanger sequencing, and then issues out a clinical report. When it comes to the sample preparation here, because our inherited disease platform really encompasses at this point around 5,000 genes. So because it's a large chunk of the genome that we want to capture, or a fairly big region, we use something called sequence capture, where we design synthetic DNA, pieces of DNA that pull down the relevant pieces of the genome, and then those relevant pieces of the genome are sequenced. Because we use these large-scale sequencers, our turnaround times for these targeted panels that we have is roughly in the order of 4 to 6 weeks. You know, the 3 is roughly our turnaround time. As we move to whole exome sequencing, we anticipate, you know, the turnaround times is going to be larger, more in the range of 3 to 4 months. At least initially when we go live. So after 2 years of work trying to figure out how to put it all together, we offered our first set of 536 genes in August of 2012. And you can see from this pie chart, our 3 major areas of focus, at least in terms of the number of genes we put in, were really neurological disorders, congenital hearing loss, congenital blindness. Those were our 3 big areas that we focused on in terms of the sample volume. There were a few metabolism genes that actually contributed quite a bit to the sample volume, but in terms of our focus and number of genes, that wasn't the area. So how we were doing in terms of sample volumes? For the first 1.5 years, where we offered the 536 gene panel, we were averaging, a little over 100 samples every 6 months. In April of 2014, we upgraded our panel from 536 to 4,800 gene panel. And that's when our volumes increased dramatically as well. Roughly on an average, we process around 300 samples every 6 months at this point in time. In terms of the specialties that utilize sequencing, from the initial group of 7 or 8 specialties that use sequencing, we now have 21 specialties that use sequencing fairly routinely. Neurology continues to be our biggest user. There's a lot of connective tissue, metabolism, and syndromic testing that also contributes substantially to our volume. And in addition, there have been, as we expanded, when we expanded from the 536 to the 4800 gene panel, we've had a lot of interest from nephrology as well in using the panels that we want, using the panels that we offer. So to give some perspective of what are the types of tests that we offer, the 4800 genes are really divided into around 250 or more test panels. These test panels range from single genes, and I think our largest test panel is around 176 or 200 genes. Now, what we do offer is flexibility in ordering. So we have these pre-made test panels that are phenotype specific that we have made in consultation with the clinicians. But we also offer the clinicians the flexibility of either ordering a part of the panel we have created as may be clinically appropriate, or in cases where the phenotype is really non-specific, we offer the option of putting together genes from various panels and creating your own custom panel. So if you look at the types of orders, so this is just the data from, and this doesn't include the data from 2016, but since we went live with the 4800 gene panel, now sequenced, close to 1800 patients. And the distribution is, there is still, the single genes still account for 25%, a quarter of all the tests we do. A lot of clinicians order the full panels, but a substantial portion, 35 to 40%, really take advantage of the flexibility that we offer and either order partial panels or custom-made panels to suit their, the clinical practice. And when we look at our total diagnostic yield, using these gene panels, the overall diagnostic yield is around 26%. So this includes the pathogenic and the likely pathogenic categories. In another quarter of the reports, we actually have a variant of unknown significance. So this really sets the stage for further testing, evaluation of family members, so on and so forth. And then half the patients, we actually don't get any results. And these results aren't terribly different from what's reported in the literature. The diagnostic yield of whole exome sequencing is roughly around the 25% range. And so we are in the same ballpark as what's been published in the literature. Now, if there is a great deal of heterogeneity by clinical diagnoses. And so the yellow bars really represent the number of tests that were ordered for particular diagnoses, and the maroon bars represent the diagnostic yield or the percent positive. So you can see, for example, Ehlers-Danlos syndrome, it's a commonly ordered test, but till date, it says 0% here, but we've had one positive case. 2 now. Matt says 2. So we have 2 positive cases of Ehlers-Danlos. On the other end of the spectrum, if you look at retinal diseases, congenital eye disorders, we have a very high diagnostic yield. We have close to 60% of the patient samples that come into our lab, we are able to provide a diagnosis. So there's a whole spectrum within that broad number of 26% that I presented before. So at this point, I'd like to just take a little bit of time and talk about the kind of work we do in the Molecular Diagnostics Lab. Because the sequencing, if you think of the workflow, a lot of the sequencing, the bioinformatics work is really done outside the context of the Molecular Diagnostics Lab. And the main, among the, when I say a turnaround time of 4 to 6 weeks, roughly 50% of the time is spent in sequencing, and the other 50% is spent in interpreting the data that we get from the sequencing output. So what is it that we do when I say variant interpretation? We think our job is really to answer the clinical question that's been posed to us. Can we identify a pathogenic variant that is causing the patient's phenotype? That's the diagnostic. Is there any significant reproductive risk? Can we identify variants that predict future health risk? And this could be in the form of secondary findings or incidental findings. And then finally, and this is not something that we do a whole lot at this point, but does the variant provide guidance for individualized management of the patient's condition? So as we progressed from small gene panels to larger gene panels, we were having growing pains in our lab. We started off with one genetic counselor, this Matt Bauer here, and he trained all the 3 pathologists who work in the Molecular Diagnostics Lab. So we pretty much didn't have a whole lot of disagreements when it came to variant interpretation. But given the growth in our sample volume, over just within the last year, we've gone from this to we have now hired one more pathologist who's going to join us in July. We went from 1 genetic counselor to 3 genetic counselors. We now have a genome analyst, and we went from 1 fellow to 2 fellows. So we were really concerned about how consistent we were going to be in interpreting genetic variants as the number of people increased. And this is a problem not just for our lab, but in the field in general. And so ACMG came up with standards and guidelines for interpreting variants. And the purpose of these guidelines really was to define categories of variants as pathogenic, likely pathogenic, all the way to the other end of the spectrum, which is benign variants. And beyond the definitions, and everybody wanted to categorize variants into categories, so very similar to these. But what these guidelines provided really was consensus on the types of evidence that can be used to make these categorizations, and also provide weights for the strength of evidence that you have for each of these categories. And at the end of the day, I think the real value of these guidelines was to try and improve the agreement amongst labs, or at the very minimum provide a framework in which we can start the discussion about how to improve agreement. And there was a follow-up paper where there were 9 sequencing labs from the CSER consortium where they used the ACMG AMP interpretation guidelines and see how well they compared across these 9 labs. And the results were kind of disappointing because they did not really improve the concordance and variant interpretation across these 9 labs. But they also noted that this guidelines provided a common framework, so when there were any discrepancies, they could go back to the ACMG guidelines and resolve those discrepancies based on the published guidelines. So at the Molecular Diagnostics Lab, what we have implemented, we've been doing this informally since the very beginning, but we have now made it a somewhat a more formal process, is that all variants really are interpreted by 2, if not 3 personnel. It's usually a genetic counselor, the fellow, and then a pathologist, all of whom look at these variants kind of independently and come to their own conclusions. And in case there is discrepancy, even even if one of the three people disagree or one of the two people disagree, then what we do is really get input from all the other peoples. So right now we have 7 people in the lab who are involved in variant interpretation. So we would like at least 4 people to agree with the interpretation that goes onto the report. The reason for this kind of approach is really data that came out from the exome sequencing project, the ESP6500 project, where the data suggested that it didn't matter how well trained or how qualified you were. Everybody made kind of the same kinds of mistakes or the equal number of mistakes. And that really developing consensus across all people, irrespective of the amount of training they had received, really improved variant interpretation. There are some caveats to that. When fellows come in July, we tend to give less weight to their interpretations, and as they progress along, their input becomes more valuable. So, but that's the general framework at this point in time. So in addition to providing all the next generation sequencing, we also provide follow-up testing or level 2 testing for family members. So in case a mutation has been identified in the proband, we provide testing of the individual mutations in family members to try and identify, well, one is for familial testing, the second is follow-up tests for variants of uncertain significance. In both these categories, we provide this level 2 test, which is a very targeted test for that particular mutation, which we provide with Sanger sequencing. In addition, our lab also provides clinical confirmation of some research results. So the International Fanconi Anemia Registry, for example, if they identify a mutation and they want to report out the results to patients, they provide us a DNA sample. We confirm the results that they have obtained in our clinical lab, and then that report goes out to the patients. The other type of alteration we now detect using next-generation sequencing data is the targeted deletion duplication analysis or copy number variation. So when you look at the type of genetic variants that are clinically relevant, CNVs are expected to occur in 12% of the genome and are implicated in pathogenesis of a variety of diseases. Currently, CNVs in most labs, are not very well detected by NGS, even though there is the theoretical promise that it can do so. There are structural variants are most commonly detected by cytogenetic mechanisms, assays. Most commonly RACGH, karyotyping, CGA, FISH, so on and so forth. But NGS does hold a promise. There are several different assays, different methods that have been developed. At last count, there were at least 16 or 17 different algorithms that have been published to look at CNVs using NGS data, at least on an exome scale. And 4 of these methods were specifically utilized in clinical labs and to detect CNVs. The problem with a lot of these methods is that we are not able to detect small CNVs. By small CNVs, I really mean under 1 kilobase. These are deletions or duplications that are really difficult to detect using NGS methods. And then the other big problem with NGS-based methods is the low specificity. Most of the published algorithms, if you look at them, demonstrate very high sensitivity, 90 to 100% in being able to detect deletions or duplications that are 1 kb or larger. But if you have, but even at the range of 1 kb or larger, the specificity is really poor. you have specificities in the range of 30% to 40% for many of these algorithms. So what that means for clinical labs is it doesn't matter whether you detect CNVs, deletions or duplications by NG-based analysis of data. The poor specificity really necessitates independent confirmation of all positive findings. So you have high sensitivity, but if you detect a CNV, that has to be confirmed by a second method. So this is now probably 4 or 5 years of work, but we took an approach where we developed our own algorithm for CNV detection using the depth of coverage that we see in CNV, that we see in next generation sequencing data. But we also added a machine learning component to that. So we added a random forest machine learning component to improve our sensitivity and specificity beyond what was done in the literature. And so this was published last year. So we basically, to provide a short synopsis, we divide our entire range of sequences into 60 base pair windows. If 3 or more windows are deleted, then we confidently call out a deletion. I shouldn't say confidently, we call out a deletion and we confirm it with a second method. If 5 or more windows are duplicated, then we call that out. And if the ratio, so we compute the coverage in the sample and we compute the coverage in a control that we run in the same run. If the ratio of the coverages is less than 0.7, call it a deletion. If it's greater than 1.4, we call it a duplication. So how did we do? When you look at just targeted panel analysis or small pieces of small number of genes, we actually showed very good sensitivity and specificity when we looked at these are 14 samples. So 14 samples having between 100 base pairs, and the largest one has an entire chromosome that was deleted. And then here, the specificity was really based on array CGH results from single genes to 13 to 14 genes. Here. So in targeted panels, we really did very well. But when we tried to scale this up without any machine learning, we identified, and I say here exome scale, but at the time we did this work, we really had only the 4,800 gene panel. And so when we scaled the CNV without any machine learning to look at exome scale data, we identified around 4 to 7 CNVs per sample, which wasn't terribly bad. But without the machine learning algorithm, we weren't able to confirm independently even a single CNV that we detected. So really poor specificity. The addition of random forest really allowed us to improve our specificity to around 50%, which is not great, but it's much better than 0%. So it went up to 50%, and our initial validation was based on 129 CNVs from 33 samples. And since then, we have done some additional work where, because it's a machine learning thing, the more data you give it, the better it starts performing. We've been able to improve the specificity on a genome-wide scale to around 75%, while maintaining a sensitivity that is close to 100%. So this is an area that is ongoing, where development is ongoing, and this is an assay that we offer currently clinically for a lot of our targeted gene panels. There are limitations to CNV-RF. I mean, because of our requirement that you need 3 consecutive windows to be deleted, the smallest deletion we can detect is 180 base pairs. and the smallest duplication we can detect is 300 base pairs. And in addition, this is not specific to CNV-RF, but regions of the genome that you cannot sequence, you cannot get any data. So there are significant areas in the genome where you can't get either variant information or sequence information, or CNV information. So in addition to address some of these limitations in CNV-RF, we also worked on a second structural variant detection algorithm. So this was called scan-indel, and here we used a variety of techniques. We used de novo assembly, where we did not assemble the sequences to a reference genome, but assembled them independently. And then we also use gapped alignment and soft clipping really to detect CNVs of various sizes, from short indels to large deletions to very large deletions in the de novo assembly. This work was really done really for our NGS oncology platform. But after we developed it, we realized that this nicely complemented the work we had done with CNV-RF, where this method was really good for detecting short indels in the ranges of between 10 base pairs to 1 kb. Scan-indel performed really well. And so we've tried to independently confirm some of the sequencing, some of the indels that have been identified by scan-indel. And so far, we've really gotten 100% concordance with scan-indel. And so that allows us, that gives us confidence that, if we detect CNVs by scan-indels, even though we are going to continue to confirm them by independent methods for now, in the future, there may be a possibility of not having to confirm all these variants by a second method. So what we are really moving towards for CNV detection as an integrated framework, where you have for large deletions, you can really use CNV-RF, especially once you go above 1 kb. This is probably a very robust method of detecting CNVs. But for the small indels, really maybe we can use a scan-indel to detect deletions and duplications up to 1 kb, and then provide an integrated report of both variants and CNVs all in a single report. So moving to the next step is I want to talk a little bit about, switch gears and talk a little bit about our oncology platform. And there are differences in how we approach design assays, short reports and stuff with regards to inherited and oncology disorders. For inherited disorders, typically the numbers of genes that you want to evaluate is very large. For oncology disorders, most of it is it's a handful of genes, I would say a few dozen genes that are really important. And when you look at the mutation distribution for inherited diseases, it's critical that we look at the entire gene. But for oncological applications, you can look at hotspots, and most of the variants that are clinically relevant and have therapeutic implications really are hotspot mutations. There is a challenge with coverage in that 20x coverage is probably enough for inherited disorders, but for oncological applications, you require very high levels of coverage so that you can detect small subclones within the tumor. And the other big areas are really the turnaround time. If you look at the turnaround time in inherited diseases, turnaround times in weeks is probably acceptable for a vast majority of the diseases. Whereas in oncological applications, you really require turnaround times within a week to be clinically relevant or useful. And then the other big thing is cost. There is, when you look at whole exome sequencing, there is a tolerance for costs, that are many thousands of dollars. Oncology, not so much. There is once you go beyond a couple of $1,000s, there is really a lot of pushback from insurance companies to be wanting to cover any of those tests. So with those differences in mind, the testing scheme we designed for oncology was a little different from inherited diseases because we were looking at small numbers of genes, and we really needed to optimize our platform for small input, small amounts of DNA input. We chose an amplicon-based enrichment using the Fluidigm platform, and I'll talk about this in a little more detail, but this was microfluidics and PCR-based enrichment. And our sequencing platform really was a desktop sequencer that's located within the Molecular Diagnostics Lab. And the main reason for that is the required turnaround time. We couldn't afford for samples to spend a week just sequencing because the report needed to be turned around in a week's time. And it came to target selection strategy. How did we select, what are the genes that we want? This is probably relevant to both oncology as well as our inherited disorders. But, we kind of conceptualized this really in the context of oncology, test panels. So on the x-axis is really the strength of evidence. On the y-axis is the size of effect. So if you had a lot of evidence, you'd move over to the right side here. If you had a very strong effect on clinical outcome, then you would be up here on the top part. So the variants that are here on the top right-hand corner are slam dunks. I mean, every clinical lab is going to do them. These are your, kind of your cancer greatest hits. Here, again, there are a lot of clinical labs which will probably include variants that fall into this category where they probably have a modest effect, not a great impact on clinical decision making, but there's a lot of studies that suggest that they are really important. The difference in the size of the panels, both for inherited disease and oncological disorders, really stem from philosophies of institutions and labs and the clinicians with respect to choice of variants in these two categories. So if you have a big size of effect, but there is not a whole lot of evidence, do you include them in a clinical panel or do you keep them as research tests? And then on this end, this is really, I would argue, belong to the research area, but depending on how many sequencers you have and the kind of institution you work in, there may be an argument for including some of these variants into a clinical test as well. So our philosophy, at least when selecting these oncology things, was really to concentrate, on these high priority variants and then focus on these areas, at least for first pass. And so we came up with a 21 gene oncology panel. 20 of them were done by NGS, and 1 gene was done just by standard sequencing. We used this Fluidigm Microfluidics Approach, and this was kind of really exciting for us when we got it. This is a microfluidics chip where you put samples on one end, you put your primers on the other end. It is in this reaction chamber in the middle where the samples and the primers come together and you get your PCR reaction. Our typical PCR reactions are usually 25, 50 microliters, but these PCR reactions are really 7 nanoliter reactions. And we have the capability of multiplexing up to 480 amplicons as a part of this chip, and we can run up to 48 samples. So you put your samples, you put your primers here, all of them get pooled and come out on this very end. We can take out those samples and put them and load them onto the sequencer and get sequencing results. So our first assay really had only 90 amplicons. We didn't maximize our throughput. 90 amplicons or 20 genes, and we were able to cover around 5,000 mutations, clinically relevant mutations that were listed in the COSMIC database. In terms of assay performance, we had optimized this assay for use of almost any type of sample that could be given to us. Our minimum DNA input really was 10 nanograms for fresh DNA, 15 nanograms for FFPE. This is in contrast if you think of our inherited disease panel. We submit approximately 500 nanograms of DNA. In inherited disease, it's not a big problem, but for oncology, it's a real challenge to get a lot of DNA from some of these samples, so we really had to optimize it for low input. Even at these low levels of 10 nanograms and 15 nanograms, up to a quarter of our samples do not meet these threshold criteria. So we have actually developed backup methods where this is more labor intensive and not as comprehensive, but we can work with DNA input as low as 1 nanogram and get sequencing results that are clinically relevant. We can detect both indels, small indels, as well as single nucleotide substitutions. And we have really validated our assay to detect mutations with a minor allele frequency of 10% or minor variant frequency of 10%. For specific hotspot mutations, we have validated down to 5%. And for indels, because there is really not a technical problem in doing them, we have validated it down to 1%. Our NGS oncology, we started it a few years later. We started in 2014. And since our start, we've been continuing to be busy. We are hoping it'll stabilize at some point, but that's not a guarantee. So probably grow, hopefully not too much. So that's what I had to talk about in terms of the assays that we do in the molecular lab. And now I'm just going to switch over to the last part of my talk, which is new developments in the Clinical Genomics Center and what we plan to do with the investment Fairview is making. A lot of the, at least a good chunk of the money that Fairview is putting towards developing the Genomics Center really goes into infrastructure development, by which I mean buying new instrumentation, hiring people, not just in the lab, but also in IT and billing and marketing and other places, to really develop the infrastructure for us to do our jobs in a more robust manner. And in 2017, in January of this year, Illumina released its latest line of sequencers called the NovaSeq. Now, the NovaSeq is probably the terminal line of sequencers in this range. And based on the specs that Illumina has provided, hopefully in the next 18 to 24 months, we believe this is the sequencer where you will get whole genome sequencing for under $1,000. And so that's the future that we have to plan for. Fairview is going to purchase one of these NovaSeq instruments. It's a $1 million instrument. All these sequences cost $1 million, give or take. So we are going to buy that instrument, but place the instrument really at UMGC instead of the clinical lab. And the idea being the capacity of this instrument is so large that our clinical samples, we don't really have a need to own a complete instrument. So placing this instrument in UMGC will allow us not only to use this instrument for clinical purposes, but also drive down the cost of sequencing for researchers at the university. Because sequencing on this instrument is going to be roughly 30% cheaper than our current instrumentation. So that's a good win-win model where we get what we need for clinical sequencing. We leverage the expertise that's already available at UMGC, and there is benefit for the researchers as well. In addition to the equipment, we also have some funds available through Fairview to develop a genomics educational program that really our goal is to try and improve familiarity with genomics for clinicians trained in specialties other than pathology. And this is an effort that's going to be led by Betsy Hirsch at our department. And we would welcome any ideas people would have about how to develop this program, because we are really just in the planning stages at this point. In terms of new assay development, we are very close to offering whole exome sequencing as a clinical test, and will likely happen in April next month. We are working on developing our integrated CNV caller, but that is probably at least a year away from clinical implementation. In addition, our NGS germline test panel, since we've been offering it since April of 2014, is woefully outdated, as many of you probably know. And so that is something that we are working on fixing as well. So our updated germline panel will be more genes, but more important, it's not going to be a lot more new panels, but the panels that we have will be updated with the genes that have been identified in the last couple of years. And then lastly, we are also working on updating our NGS oncology test. It's currently a 21 gene panel. It'll become a 62 gene panel. And the philosophy really is, in addition to genes that are clinically relevant at this point in time, we are now also including genes and variants that are in phase 2 and phase 3 clinical trials. And so if there is any potential therapeutic benefit, those genes have been added onto the panel. This is just my acknowledgement slide. None of this work would have been done without, kind of the three people who have driven all this work are Kenny Beckman at UMGC, Kevin at the Supercomputing Institute, and Matt Bauer in our lab. These are the three people who have been there from the very beginning and helped us along in this process. So there are a lot of people to thank, and Clint, who is the VP of Lab Administration, has been very supportive of this project and has really spearheaded making this a strategic priority within Fairview as well. So with that, I am happy to take any questions. Yeah.
Thank you for the interesting talk. Are there CAP surveys for the types of testing that you're doing? And if not, how do you determine proficiency?
Yeah, so CAP has developed proficiency testing both for NGS germline and NGS oncology disorders. The surveys come in many flavors. You can get sequence data, analyze the data, and send them back, just the bioinformatic analysis piece. Or you can get DNA samples, sequence it, analyze it, and send it back. In addition, so we participate in those surveys. In addition to the CAP surveys, we also exchange samples with other labs that do sequencing, next generation sequencing, and compare results across both labs twice a year. So for both NGS germline and NGS oncology, we do this semi-annually.
Thanks. So, Bharat, we see some of these kids that we know have a genetic disease. We know it's like going to be a handful of diseases. And I'm just trying to figure out what's the best way to approach these things. Do we do the sequencing and take our best shot with the first one, and then after the sequencing is done, go on to the next one and then the third one if we need to? Or do we just develop a panel for half a dozen diseases and do that? I mean, what's most cost effective and time effective?
I would argue, I mean, some of it is going to be phenotype specific. I mean, if there's a classic clinical presentation, I would say, starting small is always better because the more you sequence, the more you see things that you don't know how to interpret. That's the downside of going with large gene panels. But if you ask me in terms of, what is most cost effective, especially, I presume you're thinking more in terms of kids who do not have classic presentations, you don't actually know, which gene to go after, for example. The most cost-effective way is probably to design. When I say design, you just need to tell us what are the genes that you want in your panel, and we can put them together for you. But the most cost-effective way is probably to sequence a limited set of genes. When I say limited, most of our panels now are 25 genes or bigger. So the cost to us, the cost to us is actually the same whether we sequence a single gene or we sequence 4,800 genes. And so that's probably the most cost-effective approach. The only caveat I would give to that is we usually spend more time chasing variants of uncertain significance with larger gene panels than with smaller ones.
Well, so just to follow up with that, and maybe Sue can weigh in on this too, but for instance, we see a kid and say this kid's got Hunter or Hurler or Sanfilippo or, so there's a few number, and so we usually send off enzyme testing, which takes a while, and then, but eventually we're going to want to confirm the genotype anyway. So, at some point we may end up not messing around looking at enzyme panels and just do the genotyping. So, I don't know when we're going to cross that bridge necessarily.
Right. In some diseases, we've probably crossed that bridge already in that, like with epidermolysis bullosa, this was, we used to, and we still do, the confirmatory test is really the collagen staining, but they also require, gene sequencing. And we used to do this sequentially. Where we got the results of the collagen test first and then sent the thing out for sequencing. And now we kind of really do that in parallel, where the gene sequence, and it was more driven in terms of turnaround times and stuff like that, but we've really been doing the test in parallel, and in a lot of cases, the gene sequencing provides confirmatory diagnoses. And those results are later confirmed by collagen testing as well. So that may be a paradigm that you can consider with the enzyme testing as well. Sue, did you have a comment?
So what we found is that in certain disorders, our first line of testing is NextGen. An example, child with neonatal hyperammonemia. We can do a limited amount of biochemical testing, but we can't really easily distinguish on a rapid turnaround basis. We're going to treat them the same way. If you want a diagnosis, the probably rapidest thing is to do a selected panel.
My question involves how you choose which genes to study from the library globally. How did you choose your 4,800 or 7,000 genes, and then how does the Fairview University program compare to other genomic centers in the country in terms of number and in focus?
So our choice of how we chose our 4,800 genes and 7,000 genes was I can talk about. I think the short answer is it's driven by cost and technology. So it is a lot cheaper to buy pre-made mixes of reagents and what's available commercially drives, how much we do. That being said, our initial, when we designed our initial 536 gene panel, we really went around the university asking clinicians, what are the conditions you would like to see tested for at the university. And now that approach has morphed into really, okay, these are the set of genes that are available. Do we have all the genes we need in this panel? And what we are doing with that 7,000 gene panel is we found that the commercially available panels, set of panels, really have big holes in terms of testing for specific conditions. And so we have actually developed a supplementary capture based on our survey of what the clinicians need here at the university. Your second question about how we compare to other labs and universities, I think what differentiates us from other labs is really the breadth of testing that we offer. There are not very many labs that offer the breadth of testing we do in both germline as well as oncology disorders. And also, when it comes to germline disorders, there are really, argue, just a handful of organizations that offer gene panels that are as extensive as what we offer. And I say so. I would say we compare pretty favorably to most of the large academic centers that are there. Thank you.